Alright guys, here we are in part 1. So we're going to do some model drawing. Here is a molecule of glycine. Here's a nitrogen, 2 hydrogens on there. A carbon, 2 hydrogens on that carbon. Let me just turn it to the little bit so you can see it. 2 hydrogens on this central alpha carbon. Here's a carbonyl right here. Remember, you're supposed to draw this in your book now. This carbon has an OH attached to it. So this is a carboxylic acid. And this is the amine. This is where the name amino acid comes from. Here's your amine, that's your amino. And your acid is right there, amino acid. This is the amino acid glycine because there's 2 hydrogens on the central carbon, also known as the alpha carbon. So that's glycine. Let's switch that out for alanine. Now alanine is different. Let me see if I can make it look good for you guys on camera here. I guess that will have to do. So here we have our central carbon. There's the nitrogen attached to it with 2 hydrogens on it. So a central carbon, a nitrogen to one side, 2 hydrogens. On the other side of that central carbon, there is a carbonyl and an OH. So here's my carboxylic acid right here. Here's my carboxylic acid right here. There's my amine right there. And here is my alpha carbon, or some people call it the central carbon. It's actually called the alpha carbon. And this is alanine. How do I know? Because on the alpha carbon, which is right here, I'm pointing at it, there's a hydrogen and a carbon with 3 hydrogens. So a CH3 group, a methyl group. Remember from our naming of alkanes? So here we have an alpha carbon with a hydrogen and a methyl group on it. That makes this particular amino acid alanine. Alanine has a methyl group on the alpha carbon. Okay? Make sure you draw that neatly in your data sheet. All right, next. Glycylalanine. That essentially means, glycylalanine just means that there's a peptide with glycine first and alanine second. Now, here are just 2 amino acids bonded together, and it's already gotten extraordinarily complicated. It's already much more difficult to look at on this video. I wish you could put your hands on it. It's much easier if you can put your hands on it. But we're doing what we can during the corona crisis. So here we go. Let's take a look. Now, this is a peptide. So there's going to be a peptide bond somewhere. And it's right here. There's the nitrogen of the peptide bond. And there's the carbonyl. Remember, peptide bonds are amide bonds that connect amino acids. So here's an amino acid here. This is glycine right here. This is alanine right here. I know it's hard to see, but this is definitely alanine. So there's the nitrogen, alpha carbon, carboxylic acid of alanine, and there's alanine's methyl group right here. Here's the amide bond, the peptide bond, the amide bond. Amides are peptides. Peptides are amides, right? They connect amino acids. It's right here. There's the CH2 of glycine, and there's the NH2 of glycine. Okay, so this is glycylalanine. Now, the next drawing we're going to do is alanylglycine, where the alanine is first and the glycine is second. Let's see what that looks like. Here you go. This is alanylglycine. Now, previously, we had our peptide bond right here. On this side was alanine, or was glycine. So there's 2 hydrogens here. Now here, there's a methyl group. So there's a NH2 of alanine, the CH, the CH3. There's my amide. And then over here is glycine. There's the carboxylic acid of glycine. There's the CH2 of glycine. And here, this is the amide bond. This nitrogen belongs to the glycine. This carbonyl belongs to the alanine. This is Alanylglycine. So that's kind of interesting. We have 2 different molecules by joining 2 different amino acids. First, you can put the alanine first, then the glycine, or the glycine and then the alanine. But you have, by doing that, by just mixing the order, you have 2 different amino acids, which is probably one of the reasons why nature chose to use amino acids as one of its biggest building blocks. Why? Because you can get so much diversity from just moving the order around that you can make all kinds of different species, which I think is really kind of cool. It's important that you learn about amino acids because they're very, very important in nutrition and health sciences and all that. So do All right, guys, now we're going to do part 2 of this experiment. We're going to add acid, heavy metals, ethanol, and base to 4 of these tubes. And the 5th one we're going to use, or the first one actually, this one here, E1, we're going to put into hot water. Basically, we'll have egg white protein in each of these test tubes. And now I'm going to add hydrochloric acid to this one, and we're going to see what happens after about 5 minutes. So here we go. 1, 2, 3, 4, 5 milliliters of acid. Let's give that a little shake. Let's just wait and see what happens. Put a little base in E3. One, 2, 3, 4, 5. Put a little alcohol in E4. 1, 2, 3, 4, 5. One more for good measure. 6. And I've already put the material into E5 because I wanted to make sure it was going to work. And as you can see, it got real cloudy real fast. So let's just sit and wait. Look at E4. You can already see it starting to turn cloudy. E2, you can see at the bottom, starting to get cloudy. Let's give it a little shake. E1, we haven't done anything yet. We're going to. But you can see E4 is starting to get real cloudy. E2 is as well. So let's just let that sit there for a moment and work its magic. Find those stupid molecules. Maybe put them in the other room. I don't see them here. I think they're in the cupboard up there. There they are. Close enough. All right, guys, it's been 5 minutes, and as you can see, the acid one is definitely denaturing. If we left it for a little longer, it'd probably be a lot better. The sodium hydroxide one, surprisingly, doesn't do much. There's a little bit of denaturing here. I'm not sure you can see it on the camera. So I think it does denature. It's just not very efficient. So I would call this a positive denaturing test. This one I would probably declare negative because it really doesn't do much. The alcohol, you can see, it's definitely getting cloudier. If we left it for longer, I think it would get even more cloudy. And the heavy metal one, the one that I did off camera, you can see it's extraordinarily cloudy. So, yeah, denature, denature, negative, didn't denature, and denature. So acid denatured, base didn't seem to, ethanol certainly denatured, and heavy metal certainly denatured as well. Now, we're going to cut the video here, and we're going to go do this one together. This is test tube 1. We're going to heat this up. That's all we're going to do to it. Let's go, guys. All right, guys, here we go. Here's a test tube number E1. We're going to heat it up in this hot water. Let me turn the beaker around so we can see it. We're just going to let that heat for a couple minutes, and then we're going to see what happens to the egg albumin when we do. As you can see, it's getting much cloudier, which is expected, right? If you heat up egg white, it does turn into a soft white solid. So egg white will definitely denature in hot water like this. It's kind of how they poach eggs, essentially.